DNA is made up of a double helix of two complementary strands. The double helix describes the appearance of a double-stranded DNA, which is thus composed of two linear strands that run opposite to each other and twist together to form 6. During replication, these strands are separated. Each strand of the original DNA molecule then serves as a template for the production of its counterpart, a process referred to as semi-conservative replication. As a result of semi-conservative replication, the new helix will be composed of an original DNA strand as well as a newly synthesized strand. 7. Cellular proofreading and error checking mechanisms ensure near-perfect fidelity for DNA replication. 8. 9. In a cell, DNA replication begins at specific locations or origins of replication. 10. In the genome. 11. Which contains the genetic material of an organism. 12. Unwinding of DNA at the origin and synthesis of new strands, accommodated by an enzyme known as helicase, results in replication forks growing bidirectionally from the origin. A number of proteins are associated with the replication fork to help in the initiation and continuation of DNA synthesis. Most prominently, DNA polymerase synthesizes the new strands by adding nucleotides that complement each template strand. DNA replication occurs during the S stage of interphase. 13. DNA replication. DNA amplification can also be performed in vitro artificially, outside a cell. 14. DNA polymerases isolated from cells and artificial DNA primers can be used to start DNA synthesis. Known sequences: a template DNA molecule, polymerase chain reaction (PCR), ligase chain reaction (LCR), and transcription-mediated amplification (TMA) are examples. In March 2021, researchers reported evidence suggesting that a preliminary form of transfer RNA, a necessary component of translation, the biological synthesis, new proteins in accordance with the genetic code, could have been a replicator molecule itself in the very early development of life, or abiogenesis. 1516.